Okay. Thanks everybody for joining. Thank you, Nishal, for accepting our invitation today. So I'm gonna go get a quick introduction on Nishal. So Nishal Shah is an assistant professor in the Electrical and Computer Engineering Department at Rice University and a core member of the Rice Neuroimaging Initiative and a MacCare Medical Scholar. He did his master in electrical engineering in the Indian Institute of Technology in Delhi. And then he finished his PhD in electrical engineering from Stanford University's Artificial Retina Group in 2020. And he focused there on developing computational approaches to restore vision by electrically stimulating retinal ganglion cells. He then worked for his postdoc at Stanford's Neural Prosthetics Translation Lab and as a member of the Gate 2 Consortium until 2024, where he focused on developing communication interfaces by decoding high degrees of freedom finger movements that you're also gonna see today. So without further ado, the floor is yours, Nishal.
Yeah, thank you all for inviting me. So this work is mostly done during my postdoc and more science direction. Won't be talking about finger movement decoding as much, and that's more translational stuff. So this is all the work, part of the paper which investigates the neural geometry for multi-finger movement, simultaneous movements of multiple fingers, and this was published last year. Okay, so the finger movement is generally it's an instantiation of a more general question, which is how does the motor cortex combine single movements, individual movements, to represent multiple movements, right? And there are multiple hypotheses. So the simplest is linear summation. So in this figure on the left, I'm showing the neural state space. Each dimension is the activity of an individual neuron. And let's say this red dot is the activity corresponding to finger 1 flexion, and the green dot is finger 2 flexion. And so they, are modulated, they're represented along different directions. And when we do simultaneous flexion of both fingers like this, under a linear summation model, we would expect to get neural activity, which is over here, right? This is a very simple model. It's good because the downstream circuit, like the muscles that are controlling the fingers, have a very good, easy job of interpreting the neural activity. It can just build a decoder direction for each finger, and then that's it. And you can generalize from a subset of simultaneous finger movements to other movements, right? So it's a very simple decoder and a very simple representation. The other extreme is a non-linear combination. So in this case, simultaneous movements are represented uniquely, and they're not related in any way to the single finger movement. This is more complex, but it exploits the entire capacity of the of the neural state space, right? So the simultaneous movements are in a different direction compared to the plane spanned by finger 1 and finger 2 movement. And then there are possibilities in between. And one possibility that I'm going to talk about is there is signatures of linear summation, but then there are some non-linearities that, make it slightly more complex than the very simple linear summation model that we wanted, that we like. Okay. So in our work, we use data from two BrainGate participants, T5 and T11. And both of these participants had multi-electrode arrays called Utah arrays implanted in the hand knob region of the motor cortex, right? And in this paper, in this work, I'm going to use open loop data. So they were shown videos that look like this on the right side. And it's a virtual hand that is moving. And the trials have the standard instructed delay, task structure. So at the beginning, a marker jumps to the final position of each finger. And then after a prep period, generally around 1 or 2 seconds, the hand starts moving and the participant is instructed to just follow the movement of the hand, right? At the end, there is a hold period where they're expected to hold the finger at the end position and applying the force corresponding to either flexion or extension. Okay, and in this work, we'll be mostly looking at neural activity during the hold period, so not looking too much into the dynamics, except when I make trajectories in the neural state space. And we are using primarily threshold crossings, but uh, you know, uh, we we get the same results with with the LFP signals, or if you concatenate both the features. Okay. So let's start, and we'll first look at single finger movements and single neuron resolution, right? So the standard PSTH plots, and this is how they look like. So these are three example channels. As you can see, there is increased activity after the go period. There are some neurons which show good prep modulation. So before the go period, they have elevated firing rates. And as you can see, there is mixed selectivity, right? So individual neurons respond to different finger movements. In this case, it's responding to thumb extension and the flexion of other fingers, and it's kind of mixed selectivity. So we use the standard neural state space view, population view. And so let's start understanding the neural geometry in that space. So first, imagine this is the neural state space, and we have, this is just a cartoon. Each dot represents either finger one flexion extension or finger two flexion extension. So we first look at the local geometry, and that is characterized by the representational dissimilarity matrix. So this is just looking at the pairwise Euclidean distances between each pair of conditions during the whole period, right? And we see these two big blocks. So the extension movements across different fingers are clustered together, flexion movements are clustered together, and the thumb is slightly different. It's quite a little bit distinctly represented. To look at the global geometry, we look at the population tuning direction. So we make lines from the flexion to extension for each finger. So this is the orange line and the black line corresponding to fingers 1 and 2. And then we look at the pairwise angles between the flexion extension direction between each group of fingers, right? And this is what we get. So what we see is that the index, middle, ring, and little are all correlated to each other, with the more nearby fingers having higher correlation, and thumb is quite distinctly represented. And to understand these two plots intuitively, we make this low dimensional projection. This is using targeted dimensionality reduction. But if you do PCA, you get similar thing, except PC1 has a big CIS component. And in this case, in this plot, we are projecting the neural activity orthogonal to the subspace spanned by the condition invariant signal, right? So this is a small detail, but very important one, because some people don't remove CIS, some people subtract the CIS. In this case, we are doing a more aggressive operation where we are projecting out the subspace, which is spanned by the top three principal components of the CIS. And so what we see here is that for each finger, remember, this is single finger movement, right? So for each finger, the flexion extension direction evolve in a diametrically opposite direction. So all start from the prep, which is in the center, and during the movement, they evolve, and during the whole period, they are settling into one of the extremes. Flexion extension in different directions. And if you look at the same movement, so all the extension movements, they follow the geometry of the hand, right? Index, middle, ring, and little, with thumb being quite distinct. So this is not new. This has been shown multiple times in fMRI studies, in ECOG studies, and also intracortical multi-electrode array recordings, that the single finger movement, even after years of paralysis, as in the case of both of our participants, they follow the natural biomechanics of an able-bodied hand. Okay. So now we want to study how are the single finger movement representations combined to represent multiple finger movements, right? So first thing is, are multiple finger movements distinctly represented in neural activity? So, we have two tasks. First is this collection of 40 movements which span natural hand gestures, right? Different sign language gestures, and some of the gestures are unnatural as well, like this alternate finger gesture or the YOLO gesture. Okay. As expected from multi-electrode array recordings, we can decode individual finger movements with significant accuracy, above chance, 76%, this is good. We can now begin studying simultaneous finger movements. But there is a problem. Natural hand movements all the finger, like a lot of fingers are correlated to each other. So index and middle generally move at the same time. But to study simultaneous finger movements rigorously, we need a more, um, you know, combinatorial task design where we can decorrelate these movements so that they are not confounded, right? So we did this task where we have combinatorial movements of four finger groups, right? So in this task, we have uh, 81 different conditions. So in each condition, we have combinations of four finger movements. Each finger is either flexed, extended, or at rest. And the four finger groups are thumb, index, middle, and ring and little were combined together. We didn't do five finger groups, which is the right thing to do, but there were too many conditions to do all of them with enough repeats in a single session, right? So each finger in each condition, each finger group is independently cued to be flexed or extended or at rest. And this results in 3 raised to 4, 81 total movements. Note that this is a very hard task. There are some hand movements which are super unnatural. But kudos to our participant T5, who was very patient and very hardworking. And one thing that we did to help him was cue each movement 3 times in succession. So he can practice the first and the second time, and then the third time he can execute it accurately, in a time manner. Okay. And with this data, we get accuracy of 40%. We have so many conditions, but it's still above chance. So that's good. And so now we'll be using these two data sets, primarily the combinatorial finger movement data, to understand the geometry of simultaneous finger movements. Okay, so let's first look at pairwise movements, right? So this is a simple extension of single finger movements. So in this subset of trials, we are looking at trials where the thumb is either only the thumb is moving, either flexed here or extended, or all the other fingers which are combined, constrained to move together. So these fingers, index, middle ring, and pinky, are moving at the same time, and they're all flexed here or extended, and the thumb is at rest. And so this is the neural activity projected into the TTR space, low dimensional projection. And what we see here is that the thumb evolves around the x-axis, flexion, extension, and other fingers move along the y-axis. And now let's see where the simultaneous finger movements are, right? And so the gray lines are the combinations of these two finger groups. And this line corresponds to thumb extension and all the other finger flexion. What we observe is that the simultaneous finger movement neural activity evolves in a direction that is in between the corresponding single finger movement directions, right? So this is in between the thumb extension and all other finger flexion direction. So this is consistent with linear model, right? Linear model predicts that the neural activity should evolve in a direction which is in between the corresponding single finger movement directions. And this was reproduced in a second participant T11. And also all the other pairwise movement combinations. So here we have single finger pairs, index thumb, middle thumb, and so on. And what we see is this flower-like structure where the gray lines are always evolving in between the corresponding colored lines, single finger movement lines. Okay, so this is good, consistent with linear summation. But we have so far only looked at the direction, right? And to describe a vector, we need both direction and magnitude. And so let's look at the magnitude of neural activity. So we'll be representing the magnitude in this weird 2D plot. So this plot, we have points that correspond to the magnitude of neural activity. Remember, it's in the whole period. And so the direction of the point from the origin is the movement that was cued. So this is thumb extension positive and all the other fingers, all of the finger extension positive, right? So that's the 45-degree line. The length of the point from origin is the magnitude of neural activity. So what we can do is we can look at the magnitude of all the pairwise movement combinations, and this is how it looks like. The points along the x-axis or the y-axis are single finger movements, and the points along the diagonals 45 degree, 135 degree, and so on are the pairwise movements. And we see kind of a circle, right, where um, the magnitude is uh, following a circular shape. And now we can ask if a linear model captures this. So what we did is we took the neural activity corresponding to single finger movements, added it together, and then looked at the magnitude. And we saw that the linear model over predicts the magnitude of neural activity compared to the data. So this is a failure of the linear model, right? Linear model over predicts the neural activity magnitude, whereas the real data is more normalized. And we saw this for all the finger groups, for all the finger pairs. So these are individual finger movements, thumb index, thumb middle, and so on. And in each case, the linear model, which is the green dots here, are over predicting the neural activity compared to the data, right? So this is a failure of the linear model. And this is not, a trivial consequence of our analysis method, I would say, because in the EMG data, we collected EMG data from able-bodied participants. It shows the same flower structure in the trajectory, right, where the gray lines are in between the colored lines. But in terms of the magnitude, the linear model underpredicts the neural activity compared to the data. And this may be unique to the muscles, and there is some sort of a gain function between the neural activity and muscles so that this magnitude normalization is no longer present in muscles. Okay. Um, so now we tested this two observations that the neural activity direction for simultaneous
I have
movements
a
is
quick question,
consistent
if
with the
that's okay, before we move forward.
Yes. Yeah.
Sorry again. I was wondering if the space that you're showing us for the neural activity is a low dimensional or a high dimensional one. Seems to be a low dimensional one. And if that's the case, I was wondering which condition were used to train that lower dimension. Is it only the single fingers or all the single and the combined ones? Thanks.
Yeah, I think we use only the single finger movement directions to train the low dimensional space. Um, and one thing I would note is the visualization, we use TDR, low dimensional space. But when I'm talking about the magnitude, it's in the full neural dimensional space. And we used an unbiased estimate of the neural activity magnitude. So the magnitude analysis is not related to how we visualize the neural activity.
So for the visualization, you were taking out the condition independent out, right?
Yeah, so we take the neural activity, we project out the CIS dimensions, and then we do this targeted dimensionality reduction and then plot it. But if we do PCA, I think in the paper we have supplement figures for the PCA, and it looks similar. The flower structure is still there. It just seemed consistent to do TDR everywhere in the paper. Okay.
Thank you.
And so what is the magnitude in this same visualization space?
So visualization is a 2D space, but
Yeah.
the neural activity lives in hundreds of dimensions because we have hundreds of neurons that are recorded. And what we do is also do a pseudo population where we concatenate the neural activity from multiple sessions, right? So, and then essentially averaging the magnitude across multiple sessions.
Okay, thank you.
Can I also ask a question, Nishal, if that's okay?
Yes.
I was wondering, maybe I didn't, maybe I'm getting confused, but from your distance plots and the previous literature that you mentioned, for example, fingers like index and middle, or middle and ring pinky, they have sort of their neural trajectory. My imagination was that their neural trajectories are not really orthogonal to each other. And then in these 2D plots that I think I understood what they are, the trajectories are sort of like one of them is on the y-axis, one of them is on the x-axis, and they're not super related. For example, ring little and middle. Is that like does this mean that they're orthogonal to each other, or is this the dimensions that they're highest separated from each other, these middle and
I
big?
would say it's the latter. We are trying to look at dimensions where they're highest separated by doing TDR. That's what it would, TDR says, find me the dimensions that
Yeah,
make one finger on the x-axis and the other finger on the y-axis. Now, if
right.
you do thumb and other fingers, you see nice separation because, they're not that correlated. But
Yeah,
if you look at like index and middle, they do seem more correlated over here, right? These two colored lines are not perfectly orthogonal. Ring little, when combined together, is very strong. I mean, this is how I will present it in the, when I do the four finger combinations. So the intuition that ring little will be correlated with middle, I'm not sure if that's really true, because when you combine them as one effector, they kind of, become more dominant and can be more independent.
right. So from this we can, is this correct, that the thing that I'm gonna say, that ring little and middle, they are if you just do the full dimensional activity patterns and if during the whole time and if you correlate them, there is higher than zero correlation, for example, it's some number, but then you can find two dimensions that these two are like orthogonal from each other, for example.
Right. So, the way I think about it is when you do correlation, it matters a lot what is the origin, what do you subtract to measure correlation, right? If you're subtracting the average activity across all fingers,
Yeah.
then they will be correlated. But if you are just subtracting the average between ring, little, and middle, those two groups, they'll be less correlated, right?
Yeah.
Because from origin, everything is correlated if all the activity is on one side. But
Yeah.
when you center it, things become less correlated.
Yeah,
So it
yeah.
really depends on how you center it. When I center it to the average of um, all fingers, so like this plot, right?
Yeah.
When centering, then they look more correlated.
Right. Right.
Yeah.
So in these plots that you're showing, these are like specific to those two fingers.
Yeah. Just trying
Like,
to
yeah.
isolate the single finger movements maximally and then seeing where the pairwise movements lie with respect to that.
Yeah.
Yeah.
Thank you.
Can I also have a quick follow-up on Merta's question? So, if you were to do train the PCA on all the conditions, would you still see the complex movements living inside the same space essentially, as compared to the for example thumb or other single finger movements?
There is some dimensions that are not spanned by single finger movements, which come up in the complex finger movements. And I suspect that would be because of normalization that I talked about, right? That I'll talk about in the rest of the slides.
Right.
It's been hard to understand if that leakage into other spaces, how big is that, I guess.
Yeah, I was just like, I guess
Analysis
wondering how
we don't know in
strong is
future.
the projection to this space versus when you have all the PCs and you see those other dimensions that the complex movements are getting separated. So I guess you're saying that this effect, these projection to this single finger trained PCs are actually bigger when you do it on all the conditions. You can still see that, but there are also some dimensions that you see separation, right? But they're smaller.
Yeah, there are some dimensions that are explored which are not in the same span as single finger movement. I mean, which is natural, right? There'll always be more dimensions. Strictly linear is hard to find. But the thing is, what is trying to find some structure in the non-linear part is, I feel, interesting.
Right.
Rather than just saying that there is non-linear component, right?
Yeah. Thank you. Thank you.
Thank
Sorry.
you. We should continue
Yeah,
now.
we should.
Yeah, thanks.
Sorry.
Thank you.
Okay. Okay. So I talked about this. It's not a trivial thing because EMG doesn't have the magnitude normalization. So we try to check this in the combinatorial finger movement case. The thing is, it has more conditions, and also we are now looking at not just pairwise second order movements, but higher order movements, right? We can have up to 4 fingers moving. And so we trained a linear model, and it explains 56% of the variance. So it's not too less, but it's not too much. It's right in the middle. And these are the magnitudes of the different finger movement, contributions. You can see that the flexion modulates neural activity more on average compared to extension. And the thumb modulates the neural activity the most, and ring and little is also very strong. They're combined together, and index and middle finger, I would call them weaker modulators, weak finger movements, in this neural activity recording. So then we look at the direction, just like in the pairwise movement, we're looking how much is the direction of evolution consistent with linear summation. We can measure correlation, and linear model does better than random. So the linear model captures the direction of modulation of neural activity for combinatorial movements. And then we look at magnitude, just like what we did in the pairwise movements. And the linear model doesn't really work. It fails in a very systematic way, right? So what is this plot? Each dot is a distinct movement condition. There are 81 dots for each color, 80 dots, because one movement condition, no finger is moving, so we remove it. And then we are grouping the conditions based on how many fingers are moving, right? So this is one finger moving, single finger movements, two finger movements, three finger movements, and four finger movements. There are a lot of conditions where 2 or 3 fingers are moving as expected. And the y-axis is the magnitude of neural activity. This is the whole period activity in the full neural activity space, right? And using the cross-validated measure of distance. Now we fit a linear model to the data, and we see that the linear model captures the magnitude for 3 and 4 finger movements more. So these are conditions where 3 or 4 fingers are moving, but it really under predicts the magnitude when you have smaller number of fingers moving, right? So the linear model, there are so many conditions with 3 fingers moving and 4 fingers moving. So just because of that, the linear model matches the magnitude for these conditions. But then as a result, it under predicts the magnitude for the lower number of finger movements. Whereas in the data, the scatter plots are flat, right? So the magnitude really doesn't change if you're moving more fingers, but the linear model cannot match this dynamic range, right? Cannot either really under predicts the activity in this case for smaller number of finger movements, or for pairwise movements, as I showed before, it over predicts the activity if you use the single finger movements as the basis. Okay. And this makes sense to me because you have so many joints in the body, and we don't want the neural activity magnitude to increase as you move more and more joints, right? I mean, if you do that, then it will kind of blow up when you move your whole body, and it will be very tiny when you are moving just one finger or very single effector. So this makes sense. There should be some sort of a magnitude normalization, and a linear model cannot capture that. So we did a small extension of the linear model, and we have this multi-layered perceptron, takes the kinematics, has single hidden layer unit, and then predicting the activity of all the neurons that were recorded. And this is multiple neurons across multiple sessions. And the number of hidden units is constrained to be small, right? In this case, I'm showing 6, so much smaller than the number of neurons in the recordings. And this model predicts the variation in the magnitude, right? So the gray plots are on top of the black ones. So it captures the magnitude normalization effect. And it really does it with small number of hidden units. So the gray line is the linear model, and the red line is the linear nonlinear model. This might be a little confusing because the colors are the same as the previous plot. But as you can see, as you increase the number of hidden units, the model does better and saturates around 0.65 or something. The left one is generalizing to held out trials, and the right is generalizing to held out movement conditions, right? So you're training on subset of these 81 movements and testing it on the rest of the movements. And the nonlinear model does better with a very small number of hidden units. And how does it do it? So this last plot is looking at the cumulative distribution of the hidden unit activation. So the blue line is single finger movement. And as you increase the number of finger movements, you see more points in the histogram at -1 or +1, which is the saturation for the tan edge nonlinearity. So as you increase the number of fingers which are moving, the hidden units are just saturating, right? And that's how it's able to capture this magnitude normalization effect. So this is, our encoding model. It's just simple linear summation followed by a few elements of hidden unit nonlinearity. And I call this pseudo-linear model, extension of linear model. And now the question is, what does it mean for neural decoding, right? Because we want to ultimately build a BCI for multiple finger movements. And does this matter? Does this magnitude normalization really matter, or can we just treat everything as linear and just work? So I'll explain why this matters using a schematic. So imagine this is the neural activity space. Each dimension represents the activity of a single neuron. And this dot is the activity when you are doing flexion of a finger just by itself. And this is a dot, this is the extension for this finger. And so the line joining extension to flexion, I call this the population tuning direction for finger 1, right? Now, let's say we are moving the finger 1 along with the finger 2, which is flex, right? So let's say the finger 2 flexion modulates the neural activity along this diagonal direction. And under a pure linear summation, if I'm doing finger 2 flexion with finger 1 flexion, I will get this point in the neural activity space. It's just the summation of this and this. And if I'm doing finger 2 flexion with finger 1 extension, I'll get this point, right? This is linear summation. And now the population tuning direction for finger 1 in this different context, where the context is the other finger movement that is held constant. So this is finger 2 flexion context. This is the population tuning direction for finger 1. Because it's a linear summation model, the length of this vector and the direction of this vector are parallel to each other, right? And so under pure linear summation, there is no change in finger 1 tuning magnitude and direction across these two contexts. Now let's just do this again. But in this case, the contribution from finger 2 is much bigger in magnitude compared to finger 1 tuning direction. So this vector is much longer. And let's say there is a constraint that the neural activity cannot really exceed this ball, which is defined by biology. So the neural activity cannot exceed this magnitude. And in this case, what will happen is the linear summation will tell us that these two should be the points when you are doing simultaneous movement. But because of the magnitude constraint, you'll be projecting these points back onto the ring. And so you'll get this point over here. And then for the extension case, you get this point over here, right? And now in this second context, where the context is finger 2 flexion, if I'm measuring the population tuning direction for finger 1, I get this line, which is shorter and in a different direction compared to the finger 1 tuning direction when it was moving just by itself, right? So in this case, what happened is still linear summation, but now when we applied magnitude normalization after linear summation, there is a change in the finger 1 tuning direction, both magnitude and direction change across context, right? So what this means is if you're building a linear decoder and if the tuning direction doesn't change, then you can build a decoder from one context. So in the case on the left, we can build a linear decoder from the red line, from the isolated finger 1 movements, and then generalize it to the finger 2 movement direction, right? But that's not possible in the figure on the right, where if you build a decoder, a linear decoder for finger 1 using isolated finger 1 movements, and when you do simultaneous movement with finger 2, and there is magnitude normalization, the tuning direction changes, and we cannot really generalize the decoder, the linear decoder, from isolated movements to joint movements. Note that if there is magnitude normalization, but if the contribution of finger 2 is smaller, sorry, I have to over here, then the effect of magnitude normalization is not really, you know, observable. You don't really, you think it's really linear, but because the contribution from finger 2 wasn't really there, right? So what I'm hypothesizing is that when you are combining a strong finger of strong effector like the thumb along with a weaker effector like maybe the index finger or a middle finger, the tuning direction for the thumb doesn't really change because it's in the case in the left. But when you're combining, looking at the tuning direction for the index finger, which is a weakly represented finger in this neural population, and then you move a strong effector like the thumb, then the population tuning direction will change for this weaker effector. So we tested this in data. And so I'll be showing some of this, neural 3D projections. So first, let's look at thumb, right? So now I'm reorienting the space so that the thumb flexion extension direction is this Z direction, north-south axis, and the other fingers are along this XY plane. And then I can represent all these combinatorial finger movements with these dots. So each dot here is a single finger movement combination. All the gray dots correspond to the conditions where the thumb is flexed, and all the black dots correspond to the conditions where the thumb is extended, right? So there are 27 conditions where the thumb is flexed. There are 27 conditions where the thumb is extended. And now I'm going to join pairs of points which have the same context. So the context is, the conditions where the other fingers are doing the same thing. So for example, in this line, all the other fingers might be at rest and just the thumb is doing flexion versus extension. In this line, all the other fingers may be flexed and then the thumb is doing flexion versus extension, right? So these are different contexts and there are 27 different contexts. Okay. So now, as you can see, all these lines are kind of parallel to each other, right? So this is what we would expect from linear summation plus magnitude normalization if the effect of other fingers was weaker. And so we quantified how parallel these lines are by measuring this histogram of pairwise inner products, right? So this is the inner product between tuning directions between all these lines. A purely linear model will say that the angle should be, cosine of the angle should be 1, angle should be 0, and the linear model will just predict this point over here. And we see a spread, so it's kind of non-linear. But now we can look at how this distribution looks like across different fingers. So now we can do the same thing for the index finger, right? So now the green line is along the north-south direction, index finger flexion and extension. The gray points are all the conditions with index finger flexion. Black ones are all the conditions with index finger extension. And the lines are joining the points from the same context, right? All the other fingers are doing the same thing. And now clearly what you see is that these lines are more heterogeneous, right? They are not all in the same parallel direction like the thumb, but they're all over the place. And in the terms of the histogram is closer to 0, right? It's more heterogeneous direction. So now we can do this for all the finger groups. And we see that the middle finger, again, has different directions. And the histogram is very similar to the index finger. And the ring little, these directions are more parallel to each other, right? So we see this dichotomy. We see that thumb and ring little, which are combined together, they preserve their population tuning directions, no matter what the other fingers are doing. Whereas the index and middle, they are more susceptible to changes in their tuning direction, right? And so they're more affected by this magnitude normalization and what the other fingers are really doing. So now going back to the models, if we take a linear model, obviously the linear model says that all the 4 histograms should be peaking at 1, right? Because that's what linear model does. But now when we add a non-linearity, 6 non-linear layers, the histogram looks closer to the data. The thumb and ring little histograms, peak at a value less than 1, but still higher than the histograms for index and middle finger, right? So the tuning direction is more consistent across contexts for thumb and ring little, which are more strongly affecting the neural activity compared to index and middle fingers, which are more weakly affecting the neural activity. And so we tested this by directly looking at the generalizability of the linear decoder, right? So now what we do is, let's see if this really holds when we build the linear decoder. So in this case, we have 27 contexts. So let's say we want to build a linear decoder for what the thumb is doing. Is the thumb flexed or extended? And we can look at these different contexts. So each context is a unique combination of movements of the other fingers. And we train the decoder for thumb in one context and test it on the other context. We see that the diagonal lines values are strong on average, 92% accuracy, which is within context decoding. And so what this means is that the thumb always modulates neural activity. It's not like the thumb modulation is reducing. And across context, we see some drop. Now, if you train a decoder from one combination of finger movements for the thumb and then test it on some other combination, we see some decrease in the accuracy. But now when you look at this change between within context accuracy to across context accuracy for index and middle finger, we see that there is a much bigger drop, right? So for middle finger, it's just slightly above chance, and the off diagonal elements are less red. It's not as clean. So what this means is, if you want to build a classifier for what the middle finger position is, you cannot ignore what the other fingers are doing. So really, we need a non-linear classifier, right? Or we need to classify each of these 81 movements separately, or we need a non-linear classifier to jointly decode all the finger movements. And this generalizes to natural gestures, right? Now, we look at each dot is a single trial. X-axis is just the thumb movement, thumb flexion or extension. And then we look at pairs of natural gestures. This is a closed fist, and this is a sign language for something I don't remember. There is some changes in the tuning direction, right? So this two clouds, if you make a line between them, indicates the population tuning direction for the thumb flexion extension. And this is the population tuning direction for thumb flexion extension when all the other fingers are flexed. And the orientation changes a little bit. But if you build a classifier just using single finger movement from the x-axis, it will generalize to y-axis because the changes are not that drastic. But if you look at the little finger by itself or the ring finger by itself, these are now individually very weak. You see that the cross condition generalization is much weaker, right? So if you train it on single finger movements, it would not generalize to multiple finger movements for the little finger and ring finger. This is a linear classifier. So in summary, what hopefully I've shown is, for simultaneous finger movements, a linear model predicts the direction of neural activity, but fails to capture the magnitude normalization as more fingers are added, right? There's a magnitude normalization, which is, makes sense. The neural activity cannot blow up as more and more fingers are moving, and linear model does not capture that. And due to this, there is a context-dependent changes in the tuning direction of fingers. And mainly this affects the weakly represented fingers and not the strongly represented fingers. They are more robust and they can still be decoded along the same directions, no matter what the other fingers are doing. And the implication is that it is necessary to use non-linear decoders for brain-computer interfaces, because if you just use linear decoders, they will find one direction for each finger, and that won't work because the tuning direction really changes across fingers. And so in future, we can extend this main idea to other examples, and it has not been done before, but I'll just show some cases where it might be interesting to take this idea forward. So the first is, does this principle of linear summation and magnitude normalization extend to other multi-effector movements? So there was this nice paper by Frank Willett and Daryl Dio, where they did isolated movements of arms and legs, and then they did simultaneous movements, right? Now they were recording neural activity, incidentally from the same participant T5, as they did single and dual effector movements. And what they saw is that the arm movement direction remains consistent and the arm movement tuning remains consistent from single movements to dual movements, but the leg movement modulation decreases, right? So the leg modulation was 2.5 standard deviations, and it reduces drastically to around 1 when they do dual movements. So it's kind of the same idea on a high level. The strongly modulating effectors stay robust, no matter what the other effectors are doing, but the weakly weakly represented effectors change their tuning directions based on the context, what the other effectors are doing. And recently in human Utah recordings, people have started recording from other parts of the precentral gyrus, in addition to the hand knob region, the ventral parts that really, for the speech decoding work and so on. And the general idea is that there are hotspots. The dorsal part is hotspot for the arm movements. The ventral part is a hotspot for the facial movements or the speech movements. But there is still some tuning to the other effectors, which are not the dominant effectors for that hotspot. And this idea that the dominant effector representation stays robust in simultaneous movements, but the non-dominant effector representation changes, could be generalized to multiple, parts of the precentral gyrus. And more generally, this idea of normalization, is not new. I mean, there is a rich literature of divisive normalization, where if you're recording activity for sensory neurons like visual or auditory neurons, they change their tuning curve based on the dynamic range, right? And so if you're showing really low intensity stimulus, then the tuning curve will be over here. But then when you increase the intensity, the tuning curve will change so that we can still maximize the dynamic range within the baseline intensity. So it's very similar to that idea. And more recently, on the more neural population level, there's this nice paper that came out where they looked at prefrontal cortex activity, and they had mice doing sequences of different length. So this is a length 4 sequence and a length 5 sequence. And what they really observe is that when you increase the length of the sequence, the dynamic range remains the same, right? So in this case, this is the dynamic range, and the length 4 sequence spans this range equally. And when you change it to length 5, there are more points, there are 5 points in the same dynamic range, so they're more closely spaced apart. So it's kind of the same idea. You have limited capacity, limited magnitude, and then based on the number of things you're representing, you are dynamically allocating the dynamic range to those many things. So that's it. To summarize, simultaneous finger movements, kind of linear, but there is this big component of non-linearity coming in terms of magnitude normalization, and that could explain the apparently, apparent differences in the neural tuning for strong and weak fingers, and could be extended to many different directions within neuroscience. Thank you.
Thanks.
Thank you so much, Nishal. It was a great talk. Now questions, everyone.
I do like a higher level one. So how do you think these magnitude normalization changes throughout the practice? So let's say if the representation of the middle and the index fingers are weaker, if you train a participant on so many different types of simple and complex movements that includes those two fingers, do you think you would see, for example, that first of all, the representation of those fingers gets stronger, and you see less of a magnitude normalization on those fingers. And then you see those normalizations now, let's say, shifted towards other fingers. Or you see it gone fully, for example. How do you think about that?
Yeah, I don't know. That's an interesting question, how it will change. I mean, if you do a lot of practice, then the participant develops strategies, and they may be pushing, if they are really, yeah, I don't know the answer in short, but I would say that there could be, we just need to be careful about strategy changes with these BCI participants, right? Like if you're doing a lot of index finger, middle finger movements, then they just get better at imagining those, right? They don't really, their fingers are not moving. They're somehow relying on a connection to their fingers. And if you do a lot of practice, then their connection to these fingers becomes stronger. And I don't know how that affects neural activity in the motor cortex. It's kind of a higher level cognitive change, maybe. Who
Right.
knows?
Yeah, that makes sense.
Yeah.
Thank you.
I can ask
Go
questions.
ahead.
Thanks again, Nishal. That's very interesting, especially the result you're showing for magnitude normalization. But I was just thinking in contrast with the stuff we see in the bold signal. I don't know if you know that literature, but in the bold, at least, the more fingers you press, the larger activity in general you see. So that this contrast is very interesting to me. I was just wondering, maybe it's a dimensionality difference. For instance, yeah, Ali is telling me that it doesn't go up forever. It saturates at some point. But I was wondering that if you had a chance to look at the dimensionality of your data as more fingers are pressed, then in that sense, you can imagine that the dimensionality goes up, hence more neurons are activating or contributing, but a norm of the activity, essentially the magnitude does not change. Do you think that's a possibility?
Yeah, I haven't really looked at that too much in detail, but that's something we can look deeper. I would suspect that there will be additional dimensions that will appear when you do simultaneous movements. But it would be nice to, see if maybe some sort of participation ratio, or what is the effective dimensionality and how does that change with respect to the number of fingers. I think that will be a nice analysis. Yeah, I don't know about the different, I'm not too familiar with the bold fMRI literature. But it cannot go up infinitely.
Yeah.
So yeah, there has to be some sort of a ceiling and saturation, and that's what we see. Yeah, we should definitely look at the dimensionality closer, and like what are those new dimensions that appear, and if they are related, if they can be explained by normalize, because when you do this nonlinear, even though the magnitude normalization is a simple nonlinear operation, it could introduce new dimensions, right? It's not that it will keep the activity in the same subspace. So when I say linear summation plus magnitude normalization, it doesn't exclude the possibility that new dimensions would appear when you do combinate simultaneous movements. So both can be true.
Yeah, thank you. I also had, do you have a question? I don't know. Yeah. I was just wondering if you, I don't know, tried to connect the idea of this abstract neural activity to the complexity of the arm muscles, essentially. Meaning that, like, the idea is that essentially the four fingers, except for the thumb, are roughly controlled by one large muscle, but in the different components of that one single muscle. So in that sense, ironically, it might be actually harder to control a smaller number of the fingers rather than like a simultaneous activity of the four. In some cases, I'm not sure if that's true, but you can imagine that might happen. So I was wondering if you had any ideas of combining those models, essentially expectation of complexity under the muscle geometries to the one that you see in the neural data.
That's a very good idea. We should do it. So what you are saying is that, like, making a fist, even though it has a lot of fingers, it's a very simple movement in terms of the number of muscles, and maybe that has lower magnitude. And whereas this, or making this movement, is a very subtle movement, requires more muscles to be recruited, and that's why it has a higher magnitude, and that would explain why some 5-finger, 4-finger movements have lower magnitude and some single finger movements have higher magnitude.
Makes
Is there a is there a how do I make this notion of simple movements more concrete? Is there a metric we can use?
There are a bunch of literature on the people that have studied that, but like myosim, if you're familiar with that, essentially has a model of the arm with all the realistic muscles. For instance, you can imagine you train those models on the kind of gestures that you have, and then see what kind of muscle activity is that model producing. You can also look at the EMG and all of those. I'm sure people at some point definitely looked into that.
Yeah.
But as you exactly said, like it might be ironically harder to like individuate one movement because you now have to suppress the other ones if they are all
Right.
the same muscle. Yeah.
Yeah. Yeah, that was obviously the participant tells us that too, right? Like if I tell them to just move the middle finger or the ring finger without doing any movement of the others, or like extend that but flex one, it's hard for them to do it, and it requires more mental load.
Mm.
So yeah, so that could be a possibility for sure.
Thank you.
I can also ask one question, Nishal, if you still have time.
Sure.
It's about the model that you use, the MLP. I was wondering, so like an MLP can just fit to a lot of different complex data sets and explain it better, predict it better, and capture it better than a linear model, which is, you showed that it's very important for the control, what is it called? The BCI use case that you have. But just like thinking about it, are there explanations other than the simple, because the MLP is gonna predict your magnitude, your direction. In my head, it's gonna predict your magnitude and direction much better than a simple linear model. Is there still like some chance left for the linear model? For example, I was just thinking when reading your paper, that could it be that, for example, index and middle finger, they have this shared component? Like maybe just let's make it simple and say like there are like 100 neurons that have the shared component of middle finger extension and ring finger and index finger extension. Sorry. And then there are like unshared component, orthogonal components. And when you show the plots that these are like orthogonal to each other, you're removing the respective mean of these two. So then they become like in x-axis and y-axis, like sort of orthogonal. And then that's the non-shared component. And then the reason that you have less magnitude than simple addition of them is that when you combine these two linearly, that shared component is being counted twice when you do a simple linear model. But when you do a absolutely non-linear MLP, it's going to capture that. But then could there be like an extension of a linear model that is not fully non-linear, but also can capture this sort of magnitude saturation, other than the tangent, hyperbolic, and those things, like maybe another term to the linear model?
Yeah, that's a good idea. So what you're saying is you take 2 fingers or 2 effectors, find the shared component and the kind of private component, and then when you want to do simultaneous movement, you keep the shared component, you add it once, and the private component you add it for each finger separately, right? So it's definitely adding less. The shared component is not added twice, it's just added once, and the magnitude will be lower. It will be fun to see if that explains the magnitude completely. Yeah, this is the same thing as CIS, right? So I didn't want to add CIS multiple times, so that's why I projected it out. And so if there is one CIS that is there for all these fingers, then we have definitely taken them out. But for pairwise fingers, there could be some shared component that the CIS doesn't take out, right? And so you might still need to add it once instead of adding it twice. Yeah. Yeah, definitely. By the way, there was one model that we tried. It's in the supplement where we took the, you know, um, finger representations and normalized them before applying the linear model. So like for finger 1, it could be 1, 0. For finger 2, it could be 0, 1. And then when we want to do simultaneous finger movements, instead of doing 1, comma 1, we do 1 by root 2, comma 1 by root 2. So the magnitude of the term that goes inside the linear model stays the same.
Yep.
And this kind of make sure that you're not double counting the shared component, perhaps, but it doesn't do as well. It doesn't capture everything. But a more direct approach will be a really good idea.
Yeah, like having different parameters for those normalizations, let's say. That's, yeah. And also, like,
Yeah.
did you check if for example, in the linear model, you showed that in terms of direction, it perfectly captures the sum of like two fingers. If you, for example, if you don't calculate like r squared, and if you do r, for example, the linear model is going to perform really well, maybe close to the ceiling. I'm not sure. That's the first question. And overall, for the brain itself, and like for finger control in like brain itself, how much do you think the magnitude matters to like unentangle these activity patterns for different fingers rather than dimensionality, maybe similar to what Meredith was also mentioning.
I
Because
mean, in terms of, if you're asking the question, what is the implication for a BCI,
yeah,
decoding finger movements?
yeah,
I would just take this and say that let's just build a, non-linear
yeah, definitely,
multi-linear perceptron for decoding.
100%.
Because taking all this, basic science understanding and really coming up with a very specific decoding approach is good, but only in terms of when you have less data. When you have enough data, training data, just having a more flexible model would do the job.
Yeah.
And the takeaway for me was, when we started doing this work, this is around 2020-ish, linear models were the standard for BCI decoding. And what this said is there's a real need for nonlinear decoding. Now, the thing is, linear models are really good because when you do closed loop decoding, small errors can still be accounted for, right? And so in closed loop control, if you use a nonlinear model and get better performance on a offline setting, it doesn't mean that you will get better performance in the closed loop setting. But in this case, it's quite obvious, because if you cannot get any useful signal for the middle finger with linear decoder, then even in the closed loop setting, there is no way to rescue it. And so we need a non-linear decoder. So that was kind of the idea.
Yeah. Thank you. Thank you very much.
Okay, awesome.
I think we got to wrap it up. Let's do another round of applause for the great talk.
Thank you.
Thank you so much, NIshal, again for accepting our invitation. It was a great talk and a great paper.
Thank you.
Awesome.
That was amazing.
Yeah.
Thank
Good
you.
luck with your future works. Hope to see you soon.
Yeah.
Take care.
Take care.